afternoon. Thank you for taking the time to come to my talk today. I'm going to start my talk with a couple of invitations and end my talk with a couple of invitations. The first thing that I'd like to invite you to do is, if you're in New England, please consider coming to visit at Trinity College. I work at a college, but I have a very active research lab, and there are, at this point, nearly half of the faculty in the neuroscience program are working on the ketogenic diet, either collaborating with me or on their own. So it's really taken off at our little college. And I have many students that are very interested in this. If you're looking for any graduate students, please get in touch with me, because I'd love to keep my students in the pipeline and go further in this field. So that's one invitation. Come to Trinity. If you're in the New England area, please contact me. Another invitation that I'd like to put out to this community is I'm really hoping that we can all advocate as much as possible for brain health. It's an area of just ever increasing importance. And some of the ways that I'd like to suggest that we can advocate for brain health are through local organizations that may have a national infrastructure, and also advocate through our food policy. And from being in neuroscience for nearly 30 years now, I know this community has done more in terms of public outreach, podcasting, blogs, interconnecting, supporting each other, being very kind of pan-disease, open-minded about newcomers to the field than any other field that I've worked in, and I really think that we can do a lot if we work together. So one thing that I'm trying to advocate for is that I do think our public policies explicitly should do more to support brain health as a goal. 3 ways that I've been doing this are through the Safe Routes to School program. We know that exercise is one of the best things for your brain, and this is a national program that's executed in local communities to try and help children be able to walk or bike to school. Another organization I'm very involved in is the National Grange. It was established 150 years ago to support rural and agricultural communities and family values and economic aspects of small communities, but they have a big interest in food policy. And I think there's become a disconnect between what kind of food people want to feed their family and what kind of food policy we're supporting at the level of our government. I'm also very interested in the Trust for Public Land. They support different types of land, parks, wildlands, trails, different types of habitats all throughout the country. And being outside and having some free mental time away from our devices is also something that's very healthy for our brains. So I hope that you'll consider in your free time taking some time to advocate for brain health. So one paper that really caught my eye last year with my interest in brain health and food policy was this paper that talked about the association of higher consumption of food derived from subsidized commodities with adverse cardiometabolic risk among US adults. So probably no one in this room is surprised about that finding, but I was actually surprised that it was published in a very well-known journal, JAMA Internal Medicine. And we know that what we eat affects our brain. We've known it since the ketogenic diet was started 100 years ago, and the Charlie Foundation found that even when people switched to a more whole food diet before they went on a ketogenic diet, it could significantly reduce their seizures. So diet has a big influence on brain activity and brain health, and some of the rules that the Charlie Foundation put out for anyone are very, very simple. And it's helped people with epilepsy and many other conditions, and it's eliminate simple sugars and eat a natural whole foods diet. So really, really simple rules of the game. that could have a big impact. And if this was a bigger aspect of public policy, imagine how much better our collective brains would be. And I think that would be really impactful. So I think the title of this paper could have easily been switched to subsidized commodities are associated with adverse brain health among US adults. And there's been more and more research coming out with poor diet and increased rates of depression and anxiety, and all of these things that are very highly prevalent, not just frank neurological disorders like Alzheimer's, epilepsy, autism, etc. So there was a brain-related aspect of an accompanying commentary to the article, which the commentary was How Society Subsidizes Big Food and Poor Health. And it talked about the children of agricultural workers exposed to pesticides have IQ scores 7 points lower than those of non-exposed peers. We also know that maternal exposure to pesticides and herbicides increases the risk for autism spectrum disorder. So the comment concluded that this analysis of food subsidies points to the fact that poverty and environmental damage are public health issues, and the medical community would be valuable allies in the political coalition required to move us away from our current damaging addiction to cheap food. They are asking for our help, and we are not serving anyone if we don't advocate for what we know is the right thing. So I think this could also be changed to that these issues are brain health issues. And it makes a lot of sense. We know our brains are really metabolically expensive. So our brains take about 20% of our overall energy, even though they're only 2-3% of our overall body weight. So it makes sense that if we are not eating well, not having good metabolism, our brains are going to be kind of the canary in the coal mine. And I think epilepsy is a great disorder for this canary in the coal mine analogy because it's real-time brain activity that changes very quickly. You're not looking at development of an anatomical or structural pathology. It's real-time brain activity, which is very sensitive. So that metabolic activity is essential for maintaining brain homeostasis, which, as multiple talks have referred to, is very complicated. There's many mechanisms. And we know our brain is very adaptable and dynamic. I have this brain with the Big Bang inside because I've been very interested in evolution and how our brains evolved to become so adaptable and dynamic, how to respond to different dynamic stresses in the environment, how to learn. So that requires a lot of energy to maintain homeostasis during a lifetime of changing events. So you can look at homeostasis at many different levels, from the molecular level all the way up to the behavioral level. And I put this book up here not as an advertisement for the book, but just to show you that it exists. And this is a topic that runs through all levels of the nervous system. And to particularly mention that I was got a small amount of secret pleasure out of getting some kind of off-road topics into this book, which was supposed to be a very academic book. But I feel like if we're talking about brain homeostasis, we need to consider things like exercise, meditation, etc. So this book has chapters on those kind of fringe topics. And so another thing that I want to ask this community to advocate for is we need to do top-level science. We need to support each other. And I think we need to continue to push the boundaries. So now I'd like to get to my main topic of ketogenic diet, autism, and adenosine, three things that I care very much about. And if you haven't heard of adenosine before, you probably have been unknowingly regulating your adenosine levels in your brain, probably for years. About 80% of us ingest some kind of caffeine-containing substance, and caffeine is an adenosine receptor antagonist, and that's a psychoactive effect of blocking adenosine receptors with caffeine. So it's the most widely used psychoactive substance worldwide. Adenosine is also related to cell energy. So adenosine triphosphate, in the extracellular space, ATP is successively dephosphorylated into adenosine. And adenosine isn't a neurotransmitter per se. It's called a neuromodulator, but it binds to receptors in the synapse just like a neurotransmitter. And when ATP is released into the extracellular space, it's very rapidly dephosphorylated into adenosine, which then can have pre- and postsynaptic actions. You can see here that there's a similar ring structure to caffeine and to adenosine. And adenosine is actually used now in the operating room to stop tachycardia. Adenosine injected into the bloodstream will very rapidly stop the heart, but it will restart again. So it has very powerful and reversible effects. And I was thinking when I was putting this talk together, when we were talking about subsidized foods and cardiometabolic health, I'm like, well, why is it always about the heart? It should be about the brain. And I thought, here in the OR too, it's all the heart gets the adenosine, but the reason why the adenosine is there for the heart and not the brain is because you can't inject adenosine without stopping the heart, and that's considered an unacceptable side effect. Worse than wounds that won't heal or something like that. And we haven't figured out how to use adenosine therapeutically in the brain. But what I'd like to share with you today is I think there's many, many powerful benefits of adenosine that are very pan-disease and have major homeostatic benefits. So adenosine, as I said, links metabolism and neuronal function. And it also has immediate and long-term effects. So if we've seen in several talks, there's a metabolic dysfunction associated with all of our neurological disorders. And in most cases, up until recently, we haven't really known which came first, the metabolic problem or the neuronal problem. The ketogenic diet as a treatment for epilepsy is one case we've known for 100 years that we have a neuronal problem, excessive seizures, and we can treat it with a metabolic therapy. So that's been a clear cause and effect for many, many years that I think we didn't really take advantage of until recently when this field really started to grow and move beyond epilepsy. And the evidence is coming out in other disorders like Alzheimer's that the metabolic problem comes very early in the disorder. So Dr. Dixit mentioned yesterday that we've been trying to do all these very disease-specific treatments, yet there may be an underlying metabolic problem that could be preventing these disease-specific treatments from really being as effective as we would hope that they would be. So what does adenosine do, and why is it so interesting to me and so exciting as a therapeutic strategy? So as I mentioned, it's a neuromodulator, and it regulates neuronal activity through pre- and postsynaptic receptors. It's present throughout the extracellular space. It targets several different receptor subtypes. It can essentially enhance the signal-to-noise ratio at a synapse and tamp down excitability. The main receptor subtype that I've worked on is the adenosine A1 receptor, and caffeine really targets the adenosine A1 and A2A receptors at our normally ingested doses. It's been known for many years to be a neuroprotective molecule where additional adenosine is released during brain injury or during a seizure, and it's thought to be able to reduce excitotoxicity. So if you have severe cell stress, energy is getting low, there's a net dephosphorylation of ATP, the release of adenosine will keep down the neural activity and prevent even more neural activity requiring more energy and put the cell into a death spiral. It has a number of behavioral and physiological actions as a sleep promoter, as a pain reliever, and it's very, very powerful as a seizure reducer. It will stop any type of seizure, even medically refractory seizures, pharmacoresistant seizures. But that little problem that I told you about before with stopping the heart is the reason why we've been unable to use adenosine-based therapies to help us with these chronic medical conditions. What we know now is that adenosine also has long-term effects and has the ability to modify disease. So we think that adenosine can promote and even restore homeostasis through epigenetic changes. And this is some work done by my colleague Detlef Boisson, and some work that we did together that I'm going to share with you today. Adenosine is a kind of pan-disease link between metabolism and neuronal activity that can have both short-term and long-term effects to help get our nervous system back into balance. So I didn't know anything about ketogenic diets. I was looking for a way to regulate adenosine in the brain so that we could do those things that I knew adenosine could do if we could just get around the side effects of adenosine. So I was doing many, many experiments trying to understand the regulation of adenosine and understand brain-specific regulation of adenosine. And I won't go into, those experiments are all published, I won't go into the details. But I came up with a hypothesis that ketone-based metabolism could increase adenosine. I was kind of looking for a central mechanism that would influence adenosine. So I thought this was really exciting, really important. I didn't even know at that time that a ketogenic diet was used to treat epilepsy. So my life has really changed since I developed that hypothesis, realized that a ketogenic diet treated epilepsy, and so does adenosine, realized that a ketogenic diet treats refractory epilepsy, and so does adenosine, realized that a ketogenic diet increases ATP and improves mitochondrial function, and ATP is provides a pool of adenosine, and these are also homeostatic metabolic benefits. So after failing at getting any grants, despite many, 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 many attempts, I published this hypothesis paper in Trends in Neurosciences in 2009. They wanted me to write a paper about ATP and adenosine and glia, and I told them I wanted to do another topic, so I did this one. And it was supposed to be a review, and they got it and they said, well, this isn't really a review, this is a hypothesis paper. And we didn't ask you to write a hypothesis paper. And I said, well, it's not really a hypothesis, it's just an explanation. So ultimately, after a little back and forth, it was published. So I just want to let you know, if you get an invitation to write a paper and you want to write something that you think is more important, try to do a little bit of negotiating, because it might be a way to get out some really important ideas. So once I got this hypothesis, I started realizing all of the translational implications of what a ketogenic diet could possibly do. And what I've been working on in my lab primarily is epilepsy, pain, and more recently I've been spending most of my time on autism. And from what we know about caffeine, a vast epidemiological database about caffeine, which is a way of regulating the level of adenosine in the nervous system, we might expect that regulating adenosine through the ketogenic diet could be effective and without major side effects. So we already have epidemiological data from caffeine that we could potentially relate to regulation of adenosine by other means. So just briefly, some of the ways that I tested this hypothesis, and this data is all published, so I'm just going to show you some very brief highlights here. In our experiments, we looked at some of the clinical and animal literature, and we realized that reduced glucose seemed to be one of the key things related to seizure control. So in our initial in vitro experiments that we did in hippocampal slices, what we did is we reduced glucose to a physiological level, and we increased ATP, which we thought were two kind of endpoints of a ketogenic diet. We also fed a ketogenic diet to transgenic mice with altered adenosine signaling as a more direct test of this hypothesis. These mice have spontaneous seizures. We did experiments where we fed animals the diet and then studied slices in a dish, and we're calling this metabolic therapy in a dish. There's really no standard model for how to study a ketogenic diet in an in vitro preparation, and we feel like we found some key aspects to facilitate that. We looked at the effects of the diet during epileptogenesis, the disease process during which epilepsy develops, to try and look at these long-term ability of the diet to prevent disease. And we also looked at lasting effects of the diet after the diet was removed. So, So we were trying to look at really what's the holy grail with neurological disorders, which we don't have good strategies for, and that's disease prevention, or the C word, which is a good C word in this case, cure. So what we found in the literature was that with many slice protocols, they were applying different levels of acetoacetate or beta-hydroxybutyrate and doing various types of recordings. These are all in hippocampal slices. And the results using this approach were somewhat mixed. Some results were positive in terms of anti-seizure effects. Some results were negative. So the literature is somewhat mixed, and there's no real set standard approach for applying ketones in slices and really feeling confident that you're mobilizing the same metabolic effects as the diet. What we did in our initial experiments, as I said, is we stabilized or increased ATP through a whole cell patch pipette filled with ATP, and we reduced glucose to a physiological level of 3 millimolar. Typically, hippocampal slices are recorded in a supra-physiological glucose of about 11 millimolar, 10 or 11 millimolar. And that's kind of one of the voodoo things about slices that was when slices started. It's one of the components of the artificial cerebrospinal fluid. So we felt like that might be hampering the ability for people to study this metabolic therapy in a dish. And when we did this, reduced the glucose and stabilized intracellular ATP, we found positive anti-seizure effects. So I'm just going to go through these mechanisms fairly quickly. Here's our overall methods, all the stuff we had in our artificial cerebrospinal fluid. This is a standard glucose of 11 millimolar, standard ATP of 2 millimolar. And what we did is we manipulated our glucose down to either 7 millimolar or 3 millimolar, and we made our ATP range between 0.5 and 5 millimolar. So in these experiments, what we found is that just this change in glucose, we found a very rapid and reversible change in the membrane potential. We found a rapid and reversible change in the input resistance, so suggesting that there was a hyperpolarization due to ion channels in the membrane opening. We found that the changes were dependent on the ATP concentration. This may be small to see, but this goes from 5 to 0.5 millimolar. So we could change our impact by changing the intracellular ATP. And ultimately, we found somewhat of a dose response between glucose and ATP in terms of hyperpolarization. So hyperpolarization means you're less likely to have a seizure. And also in spontaneous release events recorded in the postsynaptic neuron. Through a series of pharmacological experiments, we determined the mechanisms underlying this hyperpolarization. And what we found is that when we had ATP levels that were stabilized at least a normal level or a little bit higher, and we reduced our glucose to 3 millimolar, which is actually a physiological level, we found that we opened specific channels called pannexins, which release ATP into the extracellular space. And the ATP in the extracellular space is very rapidly dephosphorylated into adenosine. That adenosine targets adenosine receptors, and under these conditions, we found that adenosine receptors opened a specific type of potassium channel called a K-ATP channel. Under normal conditions, adenosine receptors are coupled to a different type of potassium channel. So this was a very interesting set of mechanisms that we found. The upshot of it is that when we imposed this metabolic condition that we felt was mimicking some of the key effects of a ketogenic diet, we found this hyperpolarization and stabilization in these CA3 pyramidal cells in our hippocampal slices. These were normal rats. They didn't have epilepsy. And it was essentially an autocrine regulation of these neurons. So the neurons were regulating their own activity and almost regulating their own homeostasis. And when we looked at a little more broadly, papers that used an approach of reducing glucose from our lab, Christina Simeones, Chris Bao, Carl Stafstrom, they all had a positive anti-seizure effect. So we feel that this reduced glucose is a critical aspect for in vitro effects of the diet. But after that, what we did is we wanted to move this whole story into in vivo experiments. And this isn't what I feed my rats and mice. We feed them kind of a lard-like diet, and comes in big 10-kilogram boxes. And I had a video, but it didn't work, but it shows my students having what we call the patty party, when the students have to get together and we make little softball size, or a little smaller, aliquots of diet, put it in the freezer, because when we feed the animals this diet, it can get quite soft. We freeze it and we replace the diet every day. We feed all of our animals ad libitum, and give them fresh diet every day so they have free, full access to tasty food. So my students might eat this, but my animals don't. The diet they eat is mostly lard. What we found when we fed animals the diet and then looked at hippocampal slices is that we were able to reveal mechanisms mobilized by the diet under conditions of reduced glucose. So if we kept our glucose at 11 millimolar in our solution, we saw no difference between the control diet slices and the ketogenic diet slices. When we reduced the glucose to 3 millimolar, it had no effect on the control diet slices, but it revealed an increased sensitivity to excitability in the animals that had been fed the ketogenic diet and then had their slices re-incubated in 3 millimolar glucose. So we found a reduced response when comparing the intensity of stimulus here on the left to the amplitude of the electrophysiological response. The ketogenic diet slices showed a increasing response to increased stimulation, but the upper magnitude, the maximal response was reduced in the animals that were fed the ketogenic diet. Similarly, we induced sort of an epileptiform-like activity with a GABA blocker called bicuculline. So you can see in the middle panel, in the control diet animals, the area under the curve when the slices were exposed to bicuculline is larger than in the ketogenic slices shown below. So the larger area under the curve is showing the increased excitability. And in these experiments, and in the experiments I showed you before with the patch electrode, we were able to confirm that in animals where the slices, animals that lacked adenosine A1 receptors, these effects were completely absent. So we essentially had a genetic knockout of this mechanism with the adenosine A1 receptor knockout mice. And we were also able to reverse all these effects pharmacologically with an adenosine A1 receptor antagonist, DPCPX. In the slices that I just showed you, we recapitulated all the same mechanisms that we found in our initial experiments were found in the diet-fed slices incubated in 3 millimolar glucose. So this was very exciting because we felt like we were onto making a model of the ketogenic diet, first of all in vitro, second of all one that we could feed the diet in vivo and then study it in vitro, and now we wanted to test it under true in vivo conditions. So we used the same mice I was just talking about, mice that lacked either all or 50% of their adenosine A1 receptors. And what we would expect if you have no receptors or reduced receptors, and we had published previously, is they had no response to adenosine. So you would predict, and others had shown that they had increased susceptibility to seizures, brain injury, et cetera, because they'd lost that protective and anticonvulsant effect of adenosine. And you would predict that the ketogenic diet would not be able to protect or would only partially affect these mice in terms of their seizure protection. We also tested another mouse with manipulated adenosine, an upregulation of adenosine kinase, which is a critical enzyme for regulating adenosine, and increased adenosine kinase. The kinase phosphorylates adenosine and removes it from being able to activate the receptors. So results essentially in a decreased effective adenosine, but the receptors are still there. So in this case, we would predict that the ketogenic diet would reduce seizures in these animals. And these animals that lack the adenosine A1 receptor, so reduced endogenous adenosine signaling, or reduced endogenous adenosine signaling because it's been phosphorylated, they all demonstrate spontaneous intrahippocampal seizures. They don't show seizures behaviorally, but if you put an electrode in, they have regular spontaneous seizures. And this is something I didn't know. I had worked with these mice for years. We had no idea that they were having seizures. And I'm mentioning this because seizures, I have a feeling, are a higher comorbidity in many conditions than we realize. Recent studies have shown that there's seizure comorbidity in some Alzheimer's models. There's high comorbidity of seizures with autism. And other disorders as well. And whatever the comorbidity is estimated as, it's probably higher than that because of these intracranial subclinical seizures that you can't even detect with a scalp electrode. So you could put a scalp electrode on these animals, you won't detect seizures. You have to put an electrode in the brain. What we found, just to cut to the chase with this, because this is a very exciting experiment, is that we found in the wild type mice, which don't have seizures and don't have any alteration of their adenosine receptors or adenosine system, we found essentially no effect of the ketogenic diet. And in all of our experiments in vivo, we've tried to reverse the diet by injecting glucose. The glucose injection had no effect in the wild type animals. In the animals that had the transgenic adenosine kinase and therefore lowered adenosine tone, they have about 5 seizures an hour, but the receptors are there. We predicted that the ketogenic diet would be able to stop the seizures, and it did. It caused an 88% decrease that reversed with glucose injection. And in the adenosine A1 receptor animals that either had 50% or no adenosine A1 receptors, we found exactly what we predicted. The experiments were extremely clear. We found either partial suppression or no suppression of seizures. So the diet had absolutely no effect on the same number of seizures per hour, about 5 seizures per hour in the adenosine A1 receptor knockout mice. There was no significant change. And glucose as well had no effect on the seizure frequency. So what this shows in vivo provides very strong evidence for this adenosine-based mechanism of the diet in terms of its anticonvulsant effects. And it also shows the homeostatic regulation where normal activity was unaltered by the diet. And that's something we've seen in a number of different behavioral experiments as well. So this was published a couple of years ago, and they put a burger with this EEG. We filled in the burger, but then we fixed it by taking the bun off, so it would be a ketogenic burger. And now I think what we should do, since we were talking this morning about an MCT emulsion, we could turn this into like a keto Big Mac with an like MCT emulsion special sauce, and that would be a pretty delicious meal. Dr. Westman would approve of it at his clinic. That's kind of like the key information that we had about the mechanism of adenosine being related to the ketogenic diet. I'm just going to show you a couple of slides for some experiments that we did to try and look at the more long-term effects, because these types of effects, I think, are really the promise for the ketogenic diet in reversing and preventing disease. As I said, we've been interested in homeostasis for a long time, and epilepsy, like many other disorders, is very complicated. There's many different things that cause epilepsy. There's a lot of comorbidities and things that are going on with epilepsy. And in general, we need therapies that are going to be multi-mechanism to try and impact the multiple levels of dysfunction that are occurring in these disorders, so neural, metabolic, immune, etc. And that's what makes, I think, working on the ketogenic diet and analogous strategies so exciting. And I know it doesn't fit into the standard NIH mechanism box where you want to propose a mechanism for a problem. That's been very difficult for diet researchers. And ironically, I think this multi-mechanism metabolic resilience is what I think is the main strength. So what we found in the past few years, work done by Detlev Boisson, is that there are epigenetic effects of adenosine, and that adenosine can prevent seizures by reducing DNA methylation and have a long-term effect on seizure progression. So in addition to its ability to hyperpolarize neurons, decrease presynaptic transmitter release, so stabilize neuronal networks, and also be in this energy interchange with ATP, it's also a biochemical regulator of DNA methylation. So to study the ability of the ketogenic diet to have anti-epileptogenic effects, we looked at two models, pentylene tetrazole kindling and pilocarpine. Both result in a progressive increase in excitability in the brain. The PTZ kindling, you give successive injections of PTZ to produce that hyperexcitability. With pilocarpine, you give one triggering injection of pilocarpine, and then over time, there's a silent period, and then spontaneous seizures will start to appear. So these are both much more clinically relevant than a lot of our other epilepsy models, which are inject some excitable agent and see if the animal has a seizure. And one thing that I found interesting when I started working on the ketogenic diet is that it doesn't work in a lot of our acute mouse models of epilepsy, which I think is, since it works very well in human epilepsy, I think that's a little bit of a lesson about how good our acute mouse models are at mimicking epilepsy as a clinical condition. So what we did is we applied controller ketogenic diet prior to the epileptogenic trigger, and then we tried to look at the progression of epileptogenesis, and then we tested for lasting effects in the absence of the diet. So could there be something that could be approaching maybe a cure or a partial reversal of the disease state? We compared ketogenic diet with valproate, kind of classic anti-epileptogenic drug and anti-seizure drug. and we measured changes in adenosine and DNA methylation. And just to cut to the chase, what we found, and this is just the PTZ data, is that when we fed the diet for 8 weeks prior to starting the PTZ injections, we slowed down the progression of the kindling response. So the highest response is a 5. So we were progressively kindling the animals to become more and more excitable every time they got the injection. And the ketogenic diet significantly slowed that progression, although the animals still did start to have seizures. I mean, we're giving them a big dose of an epileptic agent. And that's another thing about the diet, is that unlike a drug, you can't just keep on increasing the dose indefinitely. So I think the diet does have powerful effects. It may take time. There may be a limit to what it can do. So I'm not thinking that it can completely cure everything, but I think it can get us partway there in many, many cases. What we found, if you look at the bar graphs on the lower right, is that in 2 separate cohorts, after 29 days, there was reduced seizure score in the ketogenic diet treated animals. And what you can see is that after we washed out the diet for 5 days, we found that in animals we kept on the diet, we still had a reduced kindled response in the yellow bar. And in the green bar, those are animals that have been reverted to the control diet. So even though they had been taken off the diet, they still retained that anti-seizure effect. In additional experiments, we showed that our hypothesis now is that the acute effects while the diet is on board are related to adenosine. So we showed a difference in adenosine between the ketogenic diet and the control diet group, and that the lasting epigenetic effects don't require adenosine to be acutely present. So we did find, like Detlev found with adenosine treatment per se, we found decreased DNA methylation in the ketogenic diet treated groups, and other groups have replicated that as well. So I'm going to spend the last part of my talk talking about autism. And I started with an interest in autism just really kind of as a fluke out of my interest in adenosine, where I noticed that a lot of the conditions where high-functioning people with autism, like Temple Grandin, who's quite famous, would say that she felt better, were all conditions that I knew would increase adenosine, or behaviors that people with autism tend to seek out. So repetitive behaviors like rocking or spinning. Temple Grandin had invented a squeeze machine. She liked that mechanical pressure. Increased metabolic demand or neural activity. So exercise or intellectual activity seem to help people with autism. And increased temperature has been recorded to have dramatic effects in some cases with autism. So my basic research that I had started on adenosine made me think that all of these things would increase adenosine. And I thought, well, that's interesting. I wonder if adenosine could have any benefits in autism. Oops. So that was my hypothesis. So then, like Dr. Westman said this morning, I did some simple math. Well, if the ketogenic diet increases adenosine, and if adenosine improves autism, then a ketogenic diet should improve autism. So I tried to get funding for this as well, and have had no success. So all of the autism research that I'm showing you today has been unfunded. I finally did get funding for some of the other experiments that I showed you previously. So the first thing I did without any funding was I developed a customized questionnaire and mobilized it through the Interactive Autism Network, which is a network of parents with children that have been officially diagnosed with autism. And this customized questionnaire had a lot of questions that I thought would either be adenosine stimulating or adenosine neutral or decreasing activities. So I tried to make kind of matched activities like a roller coaster ride versus a merry-go-round, etc. So just to get to so this is kind of a weird little study. I usually don't even talk about it, but like I said, this is an out-of-the-box crowd, so I'm just putting it out there. So and I developed it all. The parents had no idea what the hypothesis was. And what we found was the effect size. The numbers weren't large, but the effect size was quite large. When we mobilized this survey, parents responded, took the survey. We found that there was definitely a relationship between a priori conditions that we thought would increase adenosine and reduced behavioral symptoms of autism. What I didn't know when I got this hypothesis about adenosine and autism, I didn't know that seizures were a comorbidity with autism. So that was extremely interesting, as you can imagine. I didn't know that sleep issues are associated with autism, and adenosine is well known as a sleep promoter. I didn't know about these other comorbidities of autism, like anxiety. So when I put together these, various symptoms of autism, I could see where adenosine could have a major effect, and where there'd even be very direct evidence that the ketogenic diet could help. So I've been started working very busily on autism, which you probably know is prevalent and has been increasing, affects 1 in 68 children, up to 1 in 42 boys. It's very poorly treated with pharmacotherapy. Children with autism are often treated with antidepressant or antipsychotic drugs. We don't really have any drugs to treat the core symptoms of autism. Early intervention is associated with the best outcome. Not everyone has access to that. Not every child is diagnosed promptly. And the most successful intervention right now is behavioral intervention, which requires quite a lot of time and is very expensive. Parents are very motivated to try dietary approaches. So there's a lot of literature about casein-free, gluten-free diet, Feingold diet, body ecology diet. So because of the poor efficacy of the pharmacotherapy we have, parents are very motivated. And in general, there have been a few reports of the ketogenic diet which are positive. Right now, autism was collapsed into 2 main categories of behavior. So it's diagnosed behaviorally, social communication deficits and repetitive behavior or restricted interests. It's associated with neural, metabolic, and immune dysfunction. And it has a very complex etiology where we know there's a genetic component. There's no genes that explain any more than a very, very tiny fraction of the cases of autism. We think it might be a combination of common genes that sets up a genetic predisposition. And perhaps there are other environmental factors. So I mentioned earlier the maternal exposure to pesticides and herbicides is one thing that tends to increase rates of autism. So this is like Dr. Scheck's ball of yarn. There's a lot of things going on, but we're diagnosing it behaviorally. So there's a real lack of a good connection between mechanisms and outcomes. It's also a spectrum. So it ranges from people who are very, very severely affected to people who are very high functioning. But if you can improve any of the comorbidities, the anxiety, the sleep issues, et cetera, any of those things, there's a chance to then be able to capitalize more on other behavioral and educational interventions. There's a very high rate of metabolic and mitochondrial dysfunction in people with autism. So that's recently been more appreciated. The rate of mitochondrial dysfunction is 500 times the rate in the normal population. And the rate of metabolic dysfunction we think is at least as high as 30% of people with autism. So there's many mechanisms. And I just want to advocate what I think a diet plus approach could really help. So the models that we've used to study autism are the BTBR mouse, which is a mouse that's a polygenic mouse. There's no single gene, but it displays all the behavioral characteristics. It doesn't have seizures. The EL mouse, which is a model of comorbid autism and epilepsy, and the maternal immune activation mouse, which is a very clinically relevant model where moms who have a viral infection during the 1st or 2nd trimester or a bacterial infection during the 2nd trimester have an increased risk of having a child with autism. So this is a model that we and others have been using in the laboratory. And this maternal immune activation increases autistic symptoms in offspring of mice, rats, and also of monkeys. So it seems like a very translationally valid and clinically relevant model. So just to share with you some data on the poly-IC model, which was just accepted for publication like a few days ago, finally. We inject the pregnant moms with the poly-IC, which is essentially generates an immune reaction but no specific infection in the mothers. And then we did a number of different behavioral tests to look at their sociability and repetitive behavior, and I'll show you those in a second. And we tested male and female mice, and we fed them controlled or ketogenic diet, and we've done a number of different manipulations with these autism models. So what we found is that we got the expected hallmark blood changes associated with the ketogenic diet in the maternal immune activation offspring. There were no sex differences in the blood hallmarks, but using male and female animals is something we've been working on a lot in my lab the past few years to try and look at changes in estrus, changes in glucose over estrus, and try and find out if there are differential effects in females. We looked at self-grooming. These animals are actually pretty fastidious and have very complicated grooming routines. And so what we do is we videotape all of our behavior. We train everyone to be a validated scorer of behavior. And then all of our videos are scored offline, where people are blind to the diet condition. What we found is that the repetitive behavior, so self-directed grooming, what you can see in the blue bars is the the normal animals on a control diet have very low levels of grooming in the males. When we fed them a normal diet with the poly-IC, the green, there was a significant increase in the self-directed grooming, and that was mitigated in the yellow bar by feeding the animals the ketogenic diet. So that effect was found only in the males. I forgot to mention that this maternal immune activation doesn't produce autistic symptoms in females, so it's also clinically relevant in that respect where it has a differential impact on the male offspring. We also did sociability tests using a 3-chamber test of sociability, where you want to see how much time is an animal going to spend in a chamber by itself or with another mouse. So they're able to, we put mice in little jails. These are our little test mice. They're the friends that the mouse is going to meet. And then we let the mouse in, we let it habituate with no mice in there, just to show it doesn't have a significant preference for one side versus the other. We let it choose between a chamber with an empty jail or a chamber with a mouse in jail. And then we let it choose between the mouse it just met and a new mouse, so social novelty. We also measure front-to-front, face-to-face contact that the mice have when they're in this experiment. So here you can see our mouse that's in jail. And it doesn't look too happy, but you know, it's okay. And then his friend comes, and they greet face to face. So this would be scored as the mouse is in the chamber with the other mouse and is exhibiting face to face social contact. And what we found, just to cut to the chase, is that this sociability is reduced in animals that are fed animals that are have exposed to the maternal immune activation. And it's restored when the animals are fed the ketogenic diet. Social contact is actually increased to levels above normal control levels of social contact, which is a very kind of unusual effect of a treatment. But we've noticed that our mice actually seem nicer. Mice are not super nice animals, but they actually seem nicer when they're fed the ketogenic diet. So this is kind of an anecdotal observation that we quantified in this experiment. This effect, there was no impact of the maternal immune activation on social novelty or sociability in the females, but it also increased social contact in the females. So that was one effect that it did have in the females, even though they weren't impaired by the maternal immune activation. It impacted grooming during social and non-social conditions, so it really reduced this increased repetitive behavior that was caused after the maternal immune activation. And that effect was also seen in females as well. So in general, we found that the ketogenic diet improves these autism spectrum disorder associated behaviors in offspring exposed to maternal activation. As I said, I think this model is very clinically relevant and translational. It also, I didn't show you all the data today, but it also improved these behaviors in the other mouse models of autism that we've studied. So now we've looked at multiple different models and found this same effect. It never worsened these behaviors. And this is something that we found in models of Huntington's disease and other disorders where we never saw a worsening of locomotor behavior or any other behaviors that we were studying. And it didn't affect normal behavior. So showing that it has a homeostatic effect where if things are okay, it's not having a big effect. If things are awry, it tends to move them more towards the normal range. So we've been puzzling together this ketogenic diet and autism story. So I'm just going to show you an overall table. And as I said, we never saw a worsening. So these puzzle pieces represent improvement in blue, no deficit originally in yellow, no improvement in green, and red to be analyzed. So we've almost completed this table, and you can see that we have a real predominance of blue, yellow, and green. Other people have found the similar effects with other genetic models of autism and valproic acid-induced model of autism. And what I wanted to point out at the bottom is that we saw no effect in our control mice in the changes in their behavior in any of these conditions. I'm going to just cut to the chase on this. We also did the reversal experiment in autism, and this is unpublished data, but what we found is that when we had animals on the ketogenic diet and improved their sociability, we found that it was maintained after switching back to a control diet for 5 days, just like we saw in the epilepsy study. And when they were on a control diet alone, we showed that they had no change over that time period. So behavioral improvements persisted after the diet cessation. So I think we need to remember that it takes time to regain brain health, homeostasis, and resilience. Metabolic therapies are safe and effective. We think that there's an increase in mitochondria promoting this resilience and an increase in adenosine signaling at the synapses. We think that there may be both these rapid effects and long-term changes introduced by epigenetic changes. Adenosine has many different effects and is mobilized by many different conditions. And the last point I want to make is I learned that metabolism was housekeeping. And I think that that was just a terrible moniker to give to the mitochondria in our energy stores. So I want to share a little image with you of if we think of our brain as a library where everything's really organized, there's a lot of information. Sometimes a book gets out of place and you find where it's out of place and you put it back where it was. But let's say your library starts to get a little disorganized. You have trouble finding information, reduced efficiency. First, you're kind of a book nut. Next thing you know, you're on the border into turning into someone who's going to be on an episode of Hoarders, where you can't find information, can't maintain basic information, things are duplicated and broken, and whoops. Pretty soon you're at a multi-system failure, pathology, and moving isn't an option because the neurons we have are the neurons we have. So they start to get compromised and we're kind of stuck. You're hard to start bailing the ship once we've got to this point. So I've already kind of gone through the things in my summary. I think we have a lot of future opportunities to study males and females, regional differences, more research is needed on cognition, behavior, and sensory processing, also on comorbidities to try improve outcomes. We should collect prospective data, give basic advice to people with neurological disorders and parents of kids with autism, like the Charlie Foundation. Will what I eat help my epilepsy? Yes. Consider adults with high-functioning autism as maybe candidates for going on the modified Atkins diet. We need to think that food is a medical therapy. We need public policies supporting better food. I wasn't going to let you off the hook without reminding you about my first message, to advocate for brain health, because it's a nonpartisan, non-denominational issue. I'd also like to invite you to submit to Frontiers and help this field win $100,000. There's a special issue of Frontiers that's open right now. It's closing in the next month. But if you have something that you're ready to publish that's related to this area, it would be just a huge, huge advertising for this field to win this $100,000 conference. So please email me if you are interested in submitting to that. I'd like to thank Detlev Boisson and Zhang Ro, two of my main collaborators on this work, the students in my lab, and particularly my awesome colleague, Dave Ruskin. I'd like to thank people for mentioning the book, which I think is a great resource for the community to know that it exists, that 75 scientists contributed to this compendium of research, and also make sure that you know that I had 4 section editors, Detlev Boisson, Dominic D'Agostino, Eric Kossoff, and Zhang Ro, so I really only did 20% of the work, even though my name is on the cover, and I'd like to thank you for your attention.